50 years after Galvani's experiments, Emile Dubois-Reymond discovered that the passage of the nerve impulse could be detected electrically. Believing that he had identified the nervous principle with electricity, he postulated that the nerve impulses was the passage of an amount of electrical fluid down the nerve fiber. By then, the total importance of the nervous system for all life functions was well established. The Vitalists celebrated the fact that electricity had again become the life force, acting through the brain and the nerves. But this happy state did not last long. Within a year, Hermann von Helmholtz had electrically measured the speed of the nerve impulses and found it to be very much slower than that of the electricity in a wire. He concluded that while the passage of the nerve impulse could be measured electrically, it was not actually the passage of a mass of electrical particles. In 1871, unhappy with his new status of electrical force, Julius Bernstein proposed an alternate chemical explanation for the nerve impulse. He believed that the ions, charged atoms of sodium, potassium, or chloride inside the nerve cell differed from the outside tissue fluid, and that this difference resulted in the nerve cell membranes being electrically charged or polarized. In Bernstein's view, the nerve impulse was a breakdown in this polarization that traveled down the nerve fiber. Accompanied by the movement of these ions across the membrane, this, he believed, was what Dubois-Reymond had measured. The Bernstein hypothesis was eagerly accepted and has since been shown to be essentially correct, not only for nerve cells, but for all cells of the body. The success of Bernstein hypothesis resulted in the dogmatic view that this type of electrical activity is the only type permitted in the body. In this view, direct electrical currents cannot exist either within the cell or outside it, and generally in externally generated electrical currents, provided these are below levels causing shock or heat, cannot have any biological effect. The Vitalists banking on the mysterious electrical force appear to have lost the battle. While there is now no doubt that Bernstein was correct and that the membrane polarization is the basis for the conduction of the nerve impulse, it did not necessarily follow that the nerve impulse is the only method of data transmission in the nerve, or that such membrane polarization is the only way that electricity can work in the body. Orthodox science, however, discarded such ideas as vitalism. By this time, the anatomists with their microscopes had found that the nerve did not actually contact the muscle, and that a space existed between it and the muscle, which came to be called the synaptic gap. The vitalists were pushed into this small space and forced to postulate that the passage of the nerve impulse across the synaptic gap was electrical. The argument remained unsettled until 1921. Then physiologist Otto Loewe proved by experiment that transmission of the nerve impulses across the synaptic gap is also chemical. Because Loewe was a research professor at the medical school I attended, it was my misfortune and that of my classmates to have to repeat his experiment in physiology laboratory. I can say unequivocally that it worked. As a result of Loewe's experiment, all traces of electricity and magnetism were firmly excluded from any functional relationship with living things. Vitalism was finally dead. However, this triumph of science was still surrounded by a mystery. Loewe was still considered a professor at the university, even though he was elderly and retired, and he often visited the physiology laboratory, telling the students about the strange events that had surrounded his successful experiment. He had, he said, been wrestling for some time with the problem of how to do the experiment. One night, he had a dream in which the exact way to do the experiment was revealed to him. Unfortunately, he awoke and he couldn't remember any details. The next night, he had the same dream, but this time he remembered it all upon wakening. He immediately went to his lab, and in a few hours, he successfully demonstrated the chemical nature of synaptic transmission. Loewe's dream ultimately led to his receiving the Nobel Prize in 1936. In his visits to the physiology lab, he would caution us that we did not know everything, that some mysteries still remained. By the turn of the century, the idea that medicine should be based totally on science had become popular. As a result, scientific medicine based upon the chemical mechanistic model was firmly established. Its conclusive proof of efficacy came in 1909 when Paul Ehrlich discovered that the cure for syphilis lay in a specific arsenic compound. Ehrlich called this a magic bullet, a chemical specifically designed to seek out and destroy the bacterium that was its target. He further predicted that for the rest of the 20th century, medicine would be characterized by the discovery of similar specific magic bullets for all diseases. As Ehrlich predicted, this concept has dominated modern medicine. The allure of the simplistic and infallible cure is as strong today as it was in Galen's time. While medical scientists had conclusively shown that neither the electricity nor magnetism played any role in living things, the physicists and engineers had not been idle. By the 1920s, they believed they had learned all there was to learn about these two forces. We were already enjoying the luxury of electric lights, courtesy of Thomas Edison, and listening to the radio. We were able to generate, transmit, and use these forces, and we understood their characteristics. A new world based on science and technology was dawning. At that time, it appeared to be firmly established that the only way an electrical current administered to the body could have any effect was its strength were high enough to produce shock or burns. Electrical force below this level simply could not have any effect. Exposure to an electromagnetic field was even more biologically tenuous. The field was steady state, direct current, or unvarying with time, such as from a permanent magnet. It could exert a moving force only to structures or particles within the body that were themselves magnetic. Since no such magnetic material existed in the body, there could be no effect from DC magnetic fields. Furthermore, while time varying, pulsing, or AC, magnetic fields could theoretically induce electrical currents within the conducting solutions of the body. These currents would be very much smaller than those required to produce shock or heat. So again, there could be no effect. As for living organisms producing external magnetic fields, as Mesmer believed, the question was too ludicrous to even consider. The physicists, biologists, and physicians were absolutely certain that the life force simply did not exist and that all living things were simply chemical machines. They knew that life was simply the result of a chance, random event between chemicals, that it would occur in a similar fashion wherever the circumstances were right. They knew that for each disease, there was a single cause and a single therapy, and that the only valid therapy was either surgical or chemical. Finally, they knew that the living organism was simply a collection of structures which worked chemically and were integrated by means of the central nervous system with no involvement of electricity or magnetism. Life had been reduced to chemical machinery. The second wrong turn had been taken. As we shall see, the new scientific revolution has shown that the whole of the body is more than the sum of its parts, that the ability of living things to heal themselves is far greater than the mechanist thought, and that electricity and magnetism are at the very basis of life. The triumph of technology. Ulrich believed that scientists would ultimately provide us with magic bullets that would cure each and every disease seemed to come true during and following the technological explosion of World War II. The antibiotic penicillin revolutionized the practice of medicine and, more importantly, provided the hope that other chemical agents might be found that could selectively produce desired effects on cancer and other degenerative diseases. The discovery of DNA as the basis of heredity strengthened the Darwinian concept of random evolution, and it diminished the stature of human beings to that of simple machines controlled by the composition of their DNA base pairs. Advances in the understanding of the body's chemical processes and in surgical techniques such as the use of artificial organs and living transplants have nearly brought to fruition the dream of Dr. Frankenstein, the cobbling together of a human being from separate parts. We are now preparing to alter our genetic material to produce a better person. The idea that artificial organs are the same as, or even better than, those we were born with is common in the popular mind. In many ways, modern medicine has gone beyond scientific medicine to a new phase, that of technological medicine based upon the applications of this technological revolution. At the same time, technology has become dominant in society and in our lives. Electromagnetism has become dominant in society and in our lives. Electromagnetism has become the dynamo of our civilization, and through its use for power and communications, we have succeeded in changing our environment more radically than ever before. Since science has totally excluded electromagnetic forces from life, we eagerly accepted these remarkable advances without even questioning their possible biological effects. The third wrong turn had been taken. This technological revolution, now 40 years old, had begun to show its defects. The medicine it has produced an increasingly complex, expensive, and inadequate. We have failed to find magic bullets for anything other than infectious diseases. And we are facing a spectrum of new diseases against which technological medicine appears helpless. The mechanistic view of life has gradually been shown to be unable to provide satisfactory explanations for the basic functions of living things. Its proponents have confused the machinery of life with life itself, and in the process have managed to learn more and more about the machinery, but less and less about life. Two revolutions are now afoot. First, patients and physicians alike have begun to explore new avenues and approach for dealing with these mounting problems. Second, scientists, both physicists and biologists, have set about constructing new paradigm that reintroduces energy back into life. In the next chapters, we will explore this new scientific revolution. We'll see how it is bringing about the rehabilitation of some ancient beliefs and how it is influencing the development of new medical therapies while at the same time revealing the unexpected and disturbing consequences of our unbounded use of electromagnetic forces. Chapter 2, The New Scientific Revolution, the Electrical Connection. A science with all answers and no more questions, trophies into more mere technology. Michael Collier, Introduction to Grand Canyon Geology. Over the past few decades, scientists have begun to realize that mechanistic concepts penetrate merely the outer layers of life, and that under this superficial cloak lie many unexplained mysteries. Recognition of these defects in the present chemical mechanistic theory have prompted questioning and experimentation. Gradually, a new scientific concept has emerged. One has brought energetic systems back into biology and has begun to explain many of these mysteries. In particular, these energetic mechanisms have been shown to be the basis of many of the underlying control systems that regulate the complex chemical mechanisms. As we move along this new pathway, a richer biological and medical philosophy is emerging. In order to understand these advances and changes that they will bring about, we will begin the mysteries of life that the chemical mechanistic concept has failed to explain, and we will see how the new biology brings us closer to understanding the reality that integrates life, energy, and medicine. Understanding growth and healing. We are all familiar with growth. Our cuts heal, our fractures mend. We watch with love and pride the growth of our children. Physicians knew early on that some wounds did not heal. Some fractures failed to mend, and some children grew abnormally. There have been countless attempts to understand this mysterious growth process, but all have failed. The advice given to doctors in training from the time of Hippocrates to the present day has been nothing can be done to speed up, encourage, or restart a failing growth process. The best one can do is to not get in the way of Mother Nature and not do anything that would impede healing. The most familiar growth process is the healing of minor wounds. If the cut on your finger is deep enough, you know that you'll be left with a scar after it heals. This results from the most common form of healing in human beings, known as fibrosis. Fibrosis is a relatively simple closing of the wound edges with a fibrous tissue called collagen, made by specialized fibroblast cells. Technological medicine explains this process simply by describing its details. The injury produces bleeding, which forms a clot that seals off the wound. The small blood platelets within the clot release a chemical called platelet-derived growth factor, which activates the DNA within the fibroblast cells in the tissues, causing them to begin making collagen fibers. As these fibers are releasing into the wound, they contract and gradually draw the wound edges closer together. At the same time, the skin cells at the edges of the cut begin dividing and making new skin cells. These migrate over the fibrous tissue and the wound heals. In this view, the healing process is strictly local. That is, it is completely unrelated to the rest of the body and could even occur in a test tube. This sounds appropriately scientific and gives the impression that we know everything there is to know about wound healing. However, a number of questions remain. What starts this process? What tells the cells exactly what to make? What stops the process when the wound is healed? Further, this is the simplest form of healing. There are others as far more complex that are completely related to the rest of the body, and for which technology has no explanation. For example, we take the growth of any embryo for granted. The fertilized egg grows into the adult organism, be it in an earthworm, shark, mouse, or human being. The fertilized egg is one single cell with the little visible organizations, yet it becomes a living organism of immense complexity composed of billions of different kinds of cells. Orthodox science tells us that the original egg contained all of the instructions coded in its DNA to produce the different types of cells that make up the adult. The formation of the different mature cell types is accomplished by the repression of all of the DNA except that which is coded for the specific cell type required. For example, the only DNA that's active in the muscle cells is that for muscle. The rest of the DNA, while still there, is repressed and inactive. In simple terms, muscle DNA causes these cells to make certain types of proteins and intracellular structures that are characteristic of what we call a muscle cell. Hence, the muscle cell has the anatomical and functional characteristics that we call muscle because of the action of the muscle DNA alone. This process is called differentiation, and on the surface it appears to explain reasonably how well embryonic growth occurs. However, living things are not just masses of different kinds of cells brought together in a lump. The cells are organized into tissues and the tissues into organs. Most importantly, these are together organized into a discrete entity, the living organism. Mechanistic science tells us nothing of how the intricacies of this organization are produced. It doesn't tell us, for instance, how the DNA knows to make muscle here, bone here, and all the remaining organs and structures in their appropriate relationships, much less how it then ties them all together with a brain and nerve system so as to create a living organism. Obviously, what science does is simply describe the growth process. What is missing is an understanding of the control system that starts this process and then regulates it so as to produce the desired result. Nothing, be it simple healing of a cut or fertilization of a human egg, will occur without the operation of some system. When I began my work on growth process, I already developed a theory of biological control systems based on concepts derived from electronics, physics, and biology. I theorized that the first living organisms, whatever their nature, must have been capable of self-repair. Otherwise, life would not have succeeded. Self-repair requires a closed-loop control system. That is, one in which a certain signal indicates injury and causes another signal to affect repair. As the repair proceeds, the injury signal diminishes, and when the repair is complete, the signal stops. Such a system does not require consciousness or intelligence, so it may be simple in nature, but indistinguishable from life itself. It also seems to me to be realistic to assume that such a system had originated. It would continue to serve the same purposes as organisms increased in complexity. Going up the evolutionary ladder from simple organisms to human being, complexity would increase in anatomical structures and chemical reactions, but underlying everything would be the ancient control systems. Therefore, while much of my work involved growth and regeneration in unusual animals such as salamanders and frogs, I theorized that what I was studying was essentially the same for all life forms, and that my findings regarding these animals would therefore be relevant to human beings as well. This chapter will present findings from my laboratory and from labs of many other scientists, setting the stage for the discussion of the applications of these findings in the sphere of human health. Over the past 40 years, science has developed a complete theory of control systems. This theory has led to our present day computers, and lately it has begun to be applied to living systems as well. We use a wide variety of electromechanical control systems to regulate many of our everyday activities, from manufacturing goods to regulating the temperatures of our homes. A common characteristic of such control is the regulation of a powerful process by a signal of very low power. For example, in the common home oil heating system, an electrical signal of only 12 volts and very low current senses the room temperature and controls the operation of the burner unit, which operates at 110 or 240 volts and several amperes of current. The end result of this action is that the temperature of the room is held fairly constant at the desired level. The new scientific revolution against the chemical mechanistic concept resulted from the applications of new physical concepts of electricity and control system theory to the problems of growth and healing. It began when investigations found that there were many electrical currents flowing in the organism, and that these appeared to be the control signals that started and regulated growth and healing. This revolution does not replace the chemical machinery so that the fascinated the mechanists. Rather, it simply shows us what to do. The switch is that turns the machinery on. Theoretically, biological control systems should be similar to such inorganic systems as a computerized home heating system. In general, a biological system could be presumed to have a mechanism for sensing an event. For example, an injury, then transmitting this information to the central processing unit, in this case, the brain. In the brain, an automatic decision would be made to initiate repair of the wound, and a stimulating or correcting signal would then be transmitted from the brain to the site of injury. As repair took place, the injury signal would be diminished, causing the stimulating signal to also diminish. System activity would cease when the repair was completed. This is a simple negative feedback control system loop. I postulated that such systems in living organisms would be very likely to be electrical in nature. The mystery of regeneration. Regeneration is one of the healing processes that the chemical mechanistic philosophy has failed to explain, is the ability not to just heal, but to completely replace missing body parts. Many lower animals have this ability. This is common knowledge, for example, that half an earthworm will become a whole earthworm if given a little time. To many scientists, this does not seem to be much of a trick. Earthworms are pretty simple things. This view does a great disservice to earthworms, which in actuality are fascinating and complex animals that exhibit astonishing behavior. The salamander is another animal with the absolutely startling ability to regenerate missing parts. On the evolutionary scale, the salamander is an amphibian that stands a notch below the frog. From our lofty perch, amphibians may seem pretty far down the ladder. However, we are both actually in the same boat, since both human beings and amphibians are classified as vertebrates, that is, animals with a backbone and bony skeleton. The salamander has the distinction of representing the basic vertebrate from the rest of the higher animals, and humans included, have been derived. The salamander's anatomy is duplicate of our own, or vice versa. While our coccyx, a few tiny bones at the base of the spine, is a poor vestige of the salamander's tail, the rest of our bodies are remarkably similar. The salamander's foreleg has the same bones, muscles, blood vessels, and nerves in the same arrangement as a person's arm. The brain and the arrangement of the nerves throughout its body are basically the same as ours, except that the thinking area of our brains is greatly expanded. The salamander's heart has 3 chambers, ours has 4. In short, the anatomical complexity and our arrangement of this animal are remarkably similar to ours. The salamander, so like us in its body plan, is capable of regrowing in full detail a foreleg, hind leg, ear, eye, up to 1/3 of its brain, almost all of its digestive tract, and much as 1/2 of its heart. Its growth control systems are so effective that also apparently prevent it from getting cancer. In theory, then, the salamander is capable of immortality unless it is eaten. In exchange for increasing complexity of structure, we have lost most of this remarkable healing ability. We speak of regenerating severed nerve fibers in the hand, but this is simply regrowth of the fibers from the undamaged cells in our brains or spinal cords. In actuality, the only true regenerative growth we still possess is in the healing of our bone fractures. No one It's no wonder that since the regenerative abilities of the salamander were first discovered by Lazzaro Spallanzani in 1768, scientists and physicians have speculated on how this remarkable property could be restored to us. The actual process of regeneration flies in the face of some very basic tenets of the chemical mechanistic dogma. The concept of the whole being simply the sum of its parts led to the reductionist idea that each organ could be removed. Placed on the lab bench, and if properly cared for, could function as well as it did when its part was of the whole organism. The organism was simply a collection of individual parts bolted together. In this view, healing processes were localized phenomena having no relationship to the rest of the total organism and resulting simply from the local factors. This view works fine for human beings because we don't have any growth processes that challenge this localized view. Observation of the salamander, on the other hand, completely refutes this idea. Cut off the right foreleg of a salamander and it will grow a new one, complete in an anatomic detail and fully related to the rest of the animal. The salamander does not make any mistakes in its regenerative growth. It does not grow a left foreleg or an upside down leg or one that is unconnected to the rest of the animal's body by the appropriate nerve, muscle, and bone connections. The end result is a fully functional, accurate anatomical copy of the original. Obviously, the process of regeneration in the salamander must be completely related to the entire remainder of the organism by some energetic mechanism that encompasses and organizes the total organism in a fashion that cannot be explained by the chemical paradigm. To watch this miracle of a limb regeneration occur in most convincing evidence I know of for the awesome and still unknown power of life, the details of regenerative growth are even more astonishing in what they reveal of the capabilities of living cells. The first thing that happens after a salamander's limb is amputated is a rapid growth of the outermost cells of the skin across the surface. A day or two later, the cut ends of the nerves in the stump grow and make an unusual connection with each skin cell called the neuroepidermal junction, NEJ. This connection is essential for regeneration, and any technique that prevents its formation will also prevent regeneration. Shortly after the formation of NEJ, a mass of primitive cells appears between the cut end of the stump and the NEJ. The mass of embryonic cells is called the blastema, and it is the raw material from which the new limb will grow. We now know that these primitive cells come from the mature cells of bone, muscle, and so on that remain in the stump and somehow return to an embryonic state. This process of rewinding the tape of embryonic growth is called dedifferentiation, and it is the key element in bringing about regeneration. When I began my experiments on salamanders, the concept of dedifferentiation was considered hearsay. The scientific thinking of the time required that the process of repressing DNA to make specific types of mature cells be considered irreversible. The reason for this always escapes me, possibly because it required that DNA could be both repressed and de-repressed as required, giving living things too great an ability to regulate their own activities. Everyone who studies regeneration could see that a mass of primitive cells formed at the cut end of a leg. The problem was in explaining how these cells had gotten there within the constraints of the dogmatic commandment that said, "Thou shalt not consider de-differentiation." Consequently, a lot of time, effort, and money went into experiments that supposedly showed the migration of primitive cells from other parts of the body to the site of injury. Despite the fact that no one was ever really successful in showing this, the doctrine that dedifferentiation could not occur remained uncontested. On this fragile basis, the additional dogma was established that human beings failed to regenerate because they did not have such a supply of primitive cells in reserve. I cannot say what made me do my first experiment on regeneration. For some reason, I decided that it would be fascinating to see whether any differences existed between the electrical currents and voltages at the amputation sites of salamanders, who would go on to regenerate the missing legs, and those of frogs who would simply heal the cuts by fibrosis. What I was going to measure was known as the current of injury, a phenomenon that had first been observed by Galvani as being present at the site of any wound in a living animal. As a result of Galvani's discreditation by Volta, this phenomenon had received little scientific attention. Everyone assumed it was of no importance. The scientific explanation for the current of injury was simply that the injured cells in the wound leaked their charged ions across the damaged cell's membranes. This explanation was based on the old Bernstein hypothesis, but no one had ever taken the time to prove that it was correct. It was simply accepted by face value. The current of injury is a direct electrical current, DC, if it is measured with a meter. The meter reading would be constant or would vary only slowly. The actual current may be composed of electrons, such as a current in a metallic wire, or ions, charged atoms moving in a conducting solution. The former type of electrical conduction is called metallic or electronic, and the latter is called ionic. The actual electrical particles that are involved, be they electrons or ions, are called charged carriers. In all cases, the flow of a DC current implies an electrical current, an electrical circuit, a closed loop in which charged carriers are produced in one place, flow through a circuit, and ultimately return to their place of origin. In a flashlight, for example, the charged carriers, in this case electrons, are produced by the battery, given by the plus terminal at the front end of the battery to the bottom of the light bulb. They then flow through the filament of the light bulb, producing light. They leave the bulb from the side of its metallic socket and return to the other end of the battery. The turning on of the switch merely completes the circuit, letting the circuit flow and producing the light. When the Bernstein hypothesis was formulated, metallic and ionic conduction were the only two types of electrical current known. Because there were obviously no metal wires in the body, it made good sense to explain everything on the basis of ions moving in a solution. We now know that there is a third type of electrical conduction, semiconduction, in which currents may be either electrons or holes, the absence of electrons, moving in a solid material. This discovery of semiconductivity is the industrial application has given us to television sets, radios, and tape recorders, and has revolutionized our use of electromagnetic energy. We will return to semiconduction later on. Regardless of the actual type of conduction, a DC current is produced only when there is a voltage that makes it move. A voltage is produced whenever there is an excess of charge carriers with respect to another point on the same object. Since the current moves in a certain direction, our meter will detect this as either a positive or negative current. Not only was this experiment fascinating, but it turned out to have also been remarkably fortunate choice. The results were dramatic and far more significant than I had expected. I had thought that salamander would simply have more electricity than the frog. What I found was that the current of injury was negative in the salamander and positive in the frog, a difference that could not be mistaken. The experiment completely refuted the theory that the current of injury was merely a byproduct of injured cells. First of all, the charged ions in the frog are the same as those in the salamander. If it were simply a matter of charged ions leaking across damaged cell membranes, the polarity of the wounds in both animals would have to be the same. Second, the current of injury in both animals could be measured for the entire time that healing was taking place. For example, about 4 weeks during the summer months, it was inconceivable that damaged cells did not either recover or die in at most a few days. The electrical currents I measured were active agents, deliberately produced and directly related to the type of growth process the animal used to heal its wound. The control system that started, regulated, and stopped healing was electrical. In the case of regeneration, the electrical current was negative in polarity. In the case of simple scarcification healing, it was positive in polarity. But these findings could not be extrapolated to the other animals. They gave us, for the first time, firm evidence that electricity was a controlling factor in healing processes. They also raised the possibility that the system organized the total body was electrical in nature and evolutionary ancient. Because it put electromagnetic energy back into biology as a controlling factor, this simple experiment is often referred to as the beginning of the new scientific revolution. The results of experiment indicated that electricity was one basic element of a control system that regulated regeneration as opposed to simple scarcification. Much more needed to be learned. That was the nature of electrical current. Where was it coming from? What is the part of a complete control system which puts to a master control and outputs that regulated growth? It appeared possible that there was some relationship between the negative current of injury in a regenerating area and the epidermal and the neuroepidermal junctions that appeared at the same time that the current began to go in negative direction. The NEJ is an oddity. It appears only in regenerating wounds and, as noted earlier, is absolutely essential for the regenerative process. Nerve fibers normally end in special sense organs, such as touch or pressure receptors, or in excitable tissues, such as muscle. They never terminate in simple skin cells. Where they could have no action, both nerve and skin are derived from the same basic cell line, the ectoderm, and it's not inconceivable that some peculiar electrical relationship exists between skin and nerve. Since the DC electrical control system had to be evolutionary ancient, was the NEJ a specific organ developed very early in evolution, whose job it was to produce the negative DC currents? If so, the next questions to ask were, how did it produce DC electrical currents and what kind were they? In 1941, Dr. Albert Szynski proposed that the new method of electrical conduction, semiconduction, played a role in living cells. It seemed to me that this was the appropriate time to look into that possibility. Semiconductivity is a property of specific materials that gave a crystal-like structure, that is, their atoms are arranged in a regular lattice-like fashion. If an atom is the lattice has an extra electron. That electron is free to move through the rest of the lattice, hopping from one atom to the next. Likewise, if an atom lacks an electron, there is then a hole in the lattice that can move in the same fashion. The currents in the semiconductors are always very small, and only very small voltages are needed to make a current flow. What Szent-Györgyi was referring to was the fact that materials such as proteins that make up the cells have such organized structures at the time he made this suggestion. The electron microscope had not yet been invented, so he did not know the true extent to which cells and intracellular structures are organized. He had known, he would likely have been more convinced of correctness of his theory. Semiconductivity should certainly have been expected to exist in living things because of their astonishing complexity of organization. For my purposes, biological semiconduction offered the advantage of being the basis for the organized electrical currents I postulated to be the missing control systems. Ionic conduction simply could not do the job. In a number of experiments, I was able to show that the DC electrical currents I was measuring from a variety of tissues, including nerve fibers, were actually semiconducting. While the salamander's NEJ is too small to test, it probably functions in the same fashion. This biological semiconductivity is still being explored and has great promise for providing increasing insight into the life processes. As a result of the interest stirred up by these experiments, many people began to make electrical measurements of the other growth processes. The findings continued to make sense. The electrical polarity of healing human bone fractures was negative, the same as in the salamander limb regeneration. All rapidly growing tissues were found to be negative in polarity. Interestingly, cancers in animals or humans always showed the highest negativity. A consequence of this research was the idea that perhaps with the right amount of electricity, we would be able to initiate a growth process. Our initial experiments on both bone growth in dogs, done in collaboration with Andrew Bassett of Columbia University, seemed to substantiate this. Large amounts of bone growth surrounded the negative electrode, while resorption of bone occurred at the positive electrode. Since this was exactly what we had predicted, we began to think that negative electrical polarity stimulates growth, while positive polarity inhibits it. Unfortunately, these ideas, originating from early experiments done in the 1960s, became so firmly accepted that they persist today, despite being shown to be oversimplistic, as will be discussed in a later chapter. I spent a long time exploring the implications of our findings, and it was a number of years before I returned to the problem of regeneration. That brought me back to it was the question of electrical currents producing dedifferentiation. It seemed to me that this was the only process that made any sense and was forbidden by science theory. So much the better. Dr. David Murray, chairman of Department of Orthopedic Surgery at SUNY Medical School, and I had been looking at the healing of fractures, something more appropriate for two orthopedic surgeons. We chose to use frogs because they were cheaper and easier to work with than rats or mice. This turned out to be a fortunate choice because we found that the frogs simply turned the blood cells in the clot that formed at the fracture site into primitive cells, which then went on to become bone cells. Frog was able to do this because its red blood cells still contain their nuclei, while those of mammals do not. This finding immediately suggested that the red cells were being dedifferentiated by the negative electricity at the fracture site. If so, we should be able to put a frog's normal red cells in a structure in a culture dish and cause them to dedifferentiate with a similar negative current from the battery. This sounds simple, but in fact, it turned out to be a difficult and lengthy experiment. I had to choose an amount of current to use, and not being very smart, I thought I would need enough to do the job. My enough turned out to be much more than needed, leading us on a long search for the right amount. What we finally found was astonishing. The frog's red cells could be dedifferentiated by electricity, but only with vanishingly small amounts, measured in billionths of amperes. Even a little more electricity simply did not work. If we had the right amount of electrical current, in less than an hour, we could watch one red corpuscle change from a cell filled with hemoglobin containing a small shriveled up nucleus to a cell with no hemoglobin and a large active nucleus. The changes in the red cell produced this way in the culture dish were identical to those we saw in the blood clot at the frog's fracture. These results were of fundamental importance in bringing about the present scientific revolution. They clearly showed that the activities of living cells could be markedly influenced only by certain levels of extremely small electrical currents. This made the entire concept of electrical control systems feasible, and it put an end to the dogma that shock or heat were the only possible biological effects of electricity. A follow-up experiment done by my colleague, Dr. Daniel Harrington, showed that the RNA of these cells was changed exactly as required for dedifferentiation. This helped me put an end to the doctrine that dedifferentiation was forbidden. A decade of our looking at living organisms and cells in the fashion had firmly restored electromagnetism to an important role in life processes and had shown the capabilities of life are far greater than mechanistic science had envisioned. The natural conclusion was that the electricity could possibly be used to control and even start human growth processes. The first clinical experiments had done on human bone fractures had failed to heal, clearly demonstrated that bone healing could be restarted. For the first time in thousands of years of medical practice, physicians could actually control a growth process by inserting the appropriate energy. Thus, the first fruits of energy medicine were obtained. Because I like to push into the unknown, I was less interested in making fractures heal than in the prospect of actually making human beings regenerate lost parts. So, with the visions of the Nobel Prize in my head, I decided to see whether negative electricity applied over long enough time could make a new leg grow at an amputation site in the rat. It is said that a good research project yields more questions than answers, and this was such a project. My colleague, Dr. Joseph Spadaro, and I found that extremely small amounts of negative electrical current administered to an amputation between the shoulder and elbow turned on a regenerative process. An actual blastema formed and grew out of the missing structures, including bone, muscle, nerve, tendons, and so on, down to the elbow joint itself. While we could not get the growth to proceed beyond this point, the experiment nevertheless proved two important points. First, electricity was clearly a stimulus to regeneration. Second, and more importantly, the instructions required to make a new leg were retained by mammals. Therefore, the growth control system required for regeneration was present in the rat, and quite likely in human beings as well. However, something was still missing, and it was quite a few years before I learned what it was. For electricity to produce an effect upon cells, there must be not only the right amount of electricity of the right polarity, but also cells that are sensitive to DC electrical currents. In other words, not all cells are the same. Application of the right polarity and the right amount of electrical current to a sensitive cell will cause it to differentiate. The same electrical factors administered to a cell that is not sensitive will have little or no effect. In the salamander, all cells of the body will dedifferentiate if exposed to appropriate negative electrical currents. In the mammal, only certain cells of the bone marrow have this capability. This is what makes us able to hear our fractures. In our rat experiments, we dedifferentiated these cells and formed a blastema. However, the number of dedifferentiated cells was only enough to form a small blastema, which was able to regrow just the missing part of the upper foreleg and no more. Since the amount of bone marrow in the human is even smaller than in the rat, there seemed to be no chance that this technique would ever be able to produce regeneration in the human. We had the growth control system that was required for regeneration, but we did not have the number of sensitive cells needed to make it work. We were able to confirm these observations not only by applying negative electrical currents through electrodes, but also by surgically producing neuroepidermal junctions in rats. We literally stumbled into this finding in the course of an experiment designed to determine what effect a nerve would have on the cells of the bone marrow. In this experiment, which was proposed by James Cullen, a student of mine, we amputated the hind limb of a number of rats just above the knee joint. We then transplanted the cut end of the main thigh nerve into the bone marrow and brought it out the cut end of the bone. In order to keep it in place, we found that we had to suture it to the skin over the bone end. 2 weeks later, we found that we had again started a regenerative process. Just under the skin that had been sutured over the cut end of the bone were new muscle, soft tissue, and nerve fibers. Since we had not applied any electrical current, this was totally unexpected. The only way it could have occurred was if the neuroepidermal junction had somehow formed and produced the necessary negative DC electrical current. We repeated the experiment, this time with control animals that had the same amputation, but no nerve suture to the skin. After a week, we measured the DC electrical potentials at the amputation sites. The control animals were electrically positive, while those that had the nerve transplanted and sutured to the skin were negative. When we examined the later with the microscope, we found hundreds of NEJs and regenerative growth. A small experiment done out of simple curiosity had enabled us to make a remarkable discovery. The neuroepidermal junction is the source of the negative DC electrical current that stimulates regeneration, but there was and still more to be learned. It seemed apparent that placing It seemed apparent that placing the nerve directly over the cut end of the bone enabled the negative DC current to contact the bone marrow cells, and the blastema that resulted had produced the regeneration we saw. I reasoned that if we did exactly the same experiment, but this time sutured the nerve to the skin on the side of the leg away from the cut end of the bone, a neuroepidermal junction should still form and produce the negative DC current. Since there were no bone marrow cells in that area, however, no growth should occur. When we did this experiment, we found that a junction did indeed form and produce the negative DC current on the side of the leg, but as expected, there was no regenerative growth. When we then had the answer to what caused regenerative growth. The neuroepidermal junctions produced a negative DC current that stimulates sensitive cells in the area to dedifferentiate and form a blastema. Part of the control system that stimulates and controls regenerative growth had been identified. However, we did not know what told the blastema where it was in relation to the rest of the animal and what it should become. We had finally solved the riddle of why the salamander can regenerate almost anything and humans can only regenerate bone. We did find that the mammals can regenerate complex structures and presumably therefore do retain at least some of the control system. The number of sensitive cells available to dedifferentiate was found to be sufficient, and only small regenerates were possible. Without the necessary large number of appropriate cells, it appeared that the idea of restoring any major parts of regeneration to the human being was dead, at least for the time being. Discovering acupuncture. In order for a growth control system to start a healing process, it has to receive a signal indicating that an injury has occurred. During the 1960s and early 1970s, it was assumed that this signal was sent to the brain by sensory nerves that supposedly produce the sensation of pain in the conscious mind. This seemed appropriately simple and in accord with current concepts of neurophysiology. Then President Nixon visited China. When one of the reporters accompanying him had to be operated on for acute appendicitis with acupuncture anesthesia, a whole new light was thrown on this idea. Acupuncture became a common subject on the evening news. At that time, I recalled that more than a decade before, I had been visited by a colonel from the Army Surgeon General's office who had asked me what I knew about acupuncture. I said that I knew only that it was Chinese technique that involved inserting needles into specific points on the skin that were supposed to block pain, but that it made no sense because the points were not located where the nerves were. When I asked him why the Army was interested in such an oddity, he replied, "It works, and we think it may be a useful technique for battlefield anesthesia." We talked a bit about how one would go about evaluating it from a scientific point of view, and I suggested that it might be worked electrically. He left promising that I would be getting a grant to investigate acupuncture. I never did, but I continued to think about acupuncture. If the Army thought it worked, there must be something to it. Nixon's visit must have stimulated the people at the National Institutes of Health because they sent out the word that they were interested in funding studies on acupuncture. This was my big opportunity, so I proposed to the NIH that I and my colleague, Dr. Maria Reichmanis, a biophysicist, look into the possibility that acupuncture system of points and meridians carry the electrical signals of injury. We would not try to prove that acupuncture did anything clinically. Rather, we would simply apply known techniques of electrical measurements to the points and meridians to see whether we could measure anything that made sense. We reasoned that if we could find that there were reproducible and significant electrical parameters associated with the points and meridians, then these would be shown to actually exist. If we found that they did exist, then we could look for whatever anatomical structures produced them and try to discover how they carried information. The NIH funded about 20 project proposals, of which ours was one. We found that about 25% of the acupuncture points on the human forearm did exist, and the way in that they had specified reproducible and significant electrical parameters and could be found in all subjects tested. Next, we looked at the meridians that seemed to connect these points. We found that these meridians had the electrical characteristics of transmission lines, while non-meridian skin did not. We concluded that the acupuncture system was really there, and that most likely operated electrically, since clinical studies had shown that relief from pain was the one result that could be reliably produced by acupuncture treatment. This system was quite likely the input route to the brain had transmitted the signal of injury. To further prove this, I proposed that I thought was an elegant concept. DC currents flowing along a conductor always diminish in intensity the further they go because the conductor has resistance to the electrical current flow. If the current in the acupuncture system was as low as that in the output or growth stimulating side of the growth control system, then it could not be expected to go more than a few inches before it would disappear completely. Engineers handled this problem in telephone cable systems by inserting boosters amplifiers along the course of the cable to boost the signal so that it can continue on its way to the next amplifier, and so on. I proposed the acupuncture points were just such booster amplifiers spaced along the course of the meridian transmission lines. Further, because we had shown that the DC in the salamander's regenerating limb came from the neuroepidermal junctions, I proposed that the acupuncture points might be the same sort of structure. This seemed to make a great deal of sense in that the skin is where the body meets the environment and where injuries occur. Logically, the detectors for injury must be located there. Since they transmit the signal of injury by DC electrical currents, metallic acupuncture needles inserted in or near such a point would produce sufficient electrical disturbance that the amplifier could not operate and pain would be blocked. We did some preliminary experiments and found that the real acupuncture points did produce DC potentials. Not only did we show that the acupuncture points and meridians really existed, but we also had a good theory of what they were and how they worked. We proposed a number of crucial follow-up experiments of NIH, but they did not renew our grant. The missing link, the internal direct current electrical control system. As a result of the DC measurements we had made up at that time, the basic outlines of complete DC electrical growth system fell into place. Input DC electrical signals carried the information that injury had occurred along the acupuncture meridians to the brain, where part of this group signals reached consciousness and were perceived as pain. The remainder went more primitive portions to the brain, where they stimulated similar output DC signals that caused the cells and chemical mechanisms at the site of injury to produce repair. This is complete closed-loop negative feedback control system, exactly as I had postulated years before. We had shown the outlines of an internal electrical control system that regulated the repair of injuries. While this reintroduced electrical energy into the living organism as a controlling mechanism, this energy was not mysterious and unknowable. Instead, it was an energy that could be measured and perhaps even manipulated by us. For this reason, a door had been opened to profoundly important biological control processes, one with implications for clinical medicine that extend far beyond the control of normal growth. Cancer, growth out of control. Unfortunately, the normal growth of the fetus and the healing of cuts and fractures are not the only kinds of growth that human beings experience. Unlike the salamander, we suffer from the incidence of abnormal growths that can destroy us. All of diseases, cancer is the most dreaded. It is like an alien invader that inaccessibly eats away at the tissues of the body. Over the past 30 years, great advances have been done by molecular biologists, and today we know much more about mechanism of cancer than ever before. Despite this, we have not made similar advances in either prevention or the treatment of this disease. The differences between normal cell growth and cancerous growth is simply in the control exerted over the growth process. Embryonic growth and adult healing are precisely regulated by control systems that ensure that growth will be appropriate for the total organism. Cancer is the unregulated growth of cells after they have escaped these controls. What are the controls? How are they exerted? What is the wrong with controls when cancer grows? It is a problem that the deficient control systems, or is that cells have changed so that they no longer respond to normal controls. Mechanism of cancer. Molecular biology has given us the answer to one of these questions. We now know that the defect in cancer is in the cancer itself. Those agents that cause cancer, either chemical carcinogens or ionizing radiation, act by changing the genetic apparatus of the cell. Once this apparatus has been changed, the cell no longer responds to the normal control systems of the body. This discovery was actually made more than 80 years ago. In the late 1800s, Martinus Willem Beijerinck, a Dutch botanist, found that he could transmit a disease of tobacco plants known as mosaic leaf by grinding up the leaves, filtering the juice through an ultrafine filter, and then injecting the juice into healthy plants. However, the filter was so fine that it kept back even the tiniest particles, including all bacteria. There was no known living so small that it could go through the filter. Regeneration was convinced that he had discovered a new form of life, which he called filterable virus, so small that even the most powerful microscopes could not see it. No one did anything about this strange discovery until 1909, when a chicken breeder showed up at the Rockefeller Institute for Medical Research in New York with a chicken that appeared to have a cancerous tumor in its breast. The chicken subsequently died, and with Dr. Peyton Rouse, a pathologist at the Institute, performed an autopsy on it. He found that the cancer actually was a sarcoma, a cancer of the connected tissue, and that it had spread throughout the chicken's body, causing its death. The Rouse ground up some of the tumor, passed it through the same kind of filter used by Beijerinck, and injected the material into a healthy chicken. This chicken was promptly developed the same kind of cancer. The Rouse was smart enough to realize that if he had said this cancer is caused by a virus, not only would no one believe him, but his reputation would be forever tarnished. So he instead referred to a virus as a tumor agent. However, Rouse's plan did not work. No one believed him anyway. And for more than 50 years, the possibility that cancer was caused by viruses was never fully explored. However, the sarcoma cells Rouse has isolated grew very well in culture, and they were propagated in this way. Throughout those years, with the discovery of the electron microscope, viruses were able to be seen, including both the tobacco mosaic viruses and the Rouse sarcoma virus.